Welcome to the UBC Neuroscience Association podcast. I'm here with Dr. Tim O'Connor, who is a professor of neuroscience in the Faculty of Medicine. He has a bachelor's and a PhD from the University of Toronto and a postdoc from UC Berkeley. Thanks for being here.
Thank you, Hriday.
Awesome. I want to get started with just sort of talking about what brought you to neuroscience, how you decided to do this, and how you ended up building a career in neuroscience.
Yeah, that's a good question. I think as like many undergraduate students where I really wasn't sure what I wanted to do in my life. I had an older brother who was in medicine at the University of Toronto at the time that I was an undergrad. And so I was thinking about the medical field, and I took the opportunity to do some research as an undergraduate student in someone's lab, Dr. Derek Vanderkooy. And it was during that period of actually doing undergraduate research that I really thought, this is really something I'd like to try. I wasn't really enamored with neuroscience at the point. I was really just enamored with my project, which happened to be, how do neurons develop, and how does our brain develop? And at first, I thought it was okay to try this, but as I got deeper and deeper into the project, I really became, in love with neuroscience. And so I would say when I started my graduate career with Dr. Vanderkooy, it was in the first year that I really realized that this is something that I want to really make a career out of.
Right on. So you entered undergrad sort of not knowing all that much about neuroscience, but generally being interested in health sciences.
Yeah, generally interested in health sciences, and I did an undergrad in Toronto. They call it a specialist, and I did that in physiology.
Gotcha.
And as part of the physiology program, we had a fair amount of neurophysiology as well. So that also was capturing my attention as well.
Very cool.
Yeah.
And then you sort of went straight into grad school after undergrad?
Yep, right into grad school. If there's one thing I would change in my life, I would say if you ever plan on traveling as a student, I think you should take that opportunity maybe before you go into grad school, certainly before you go into a postdoc, because as you progress along your career, you have less and less opportunity to take that time off. And so if there's one thing I would change was maybe to delay going to grad school by a year. But yeah, I went straight into grad school after my Bachelor of Sciences, and I went into the same lab. And I was really interested at the time, oddly enough, in motivation.
Interesting.
So yeah, what are the addictive qualities that are in things like alcohol and drugs, that motivate people to take these? But at the time, the lab was quite small, and Dr. Vanderkooy told me that he already had enough people in that area, and so he put me on a project that looked at the development of the brain. And that's when I really started to fall in love with developmental neurobiology. Yeah.
Gotcha. So you wanted to start in behavioral psychology, as it was called back then, or behavioral neuroscience now, and then sort of found your way by circumstance into developmental neuroscience.
Exactly. Yeah.
Very cool. And then how long was your PhD, and what did those years look like?
Yeah, so I was fortunate that I finished my PhD in about 4.5 years. And I had a number of papers from the PhD, and that provided me the opportunity to apply for postdoctoral fellowships. And I was lucky enough to get a postdoctoral fellowship from the Medical Research Council of Canada. And what that does is it provides more opportunities for you. And that's where I could start looking at larger labs in the States, to see where I might sort of further my training and education. But I have to say, the PhD experience was tremendous.
Right on.
Dr. Vanderkooy had a really dynamic lab. I made some lifelong friends from the lab. We worked hard, and we played hard pretty well too.
Right
so
on.
It was good. Yeah, I was be able to maintain, playing hockey and all my sports things that I like to do as well. So
tell me about that. I know you've had like this interest in hockey from a young age. What was that whole career like?
I've always had an interest. So I started playing hockey when I was a young kid, and I was the only person in my family. I have 5 siblings, but I was the only one that played hockey. And so I played throughout my life. And then when I was in grad school, I would play at the U of T. They'd have a hockey league that I played for, and I played for St. Mike's College. And when I went to do my postdoctoral fellowship at Berkeley, I played for the University of California Berkeley hockey team.
Right on.
Whereas was in my early 30s by the time I left there, and I was playing with like these 18-year-old kids that were just coming in as undergrads. So
Yeah.
it was quite the experience. And hockey is just one of those sports, like many sports, where it's more than just the activity. It's you build up lifelong friendships.
Right on.
And I still have great friends from all the way through my hockey career, including the time at UC Berkeley when I played with people from all ages. Yeah, okay. It was pretty great.
Very cool. I want to go back to sort of when you were doing your PhD. At that point, did you start thinking about sort of, I'm in grad school now, I want to go into academia? Was that like in the back of your mind?
Yeah, back at that period, there really weren't that many options. Genentech hadn't really started. Maybe it was just, the whole biotech industry was really in its nascent stages. Right. So your primary job opportunities back then was academia, and there, academia was great. I mean, universities were expanding; they were getting larger. They were hiring people, right. and there was money. I mean, when I first came to UBC, the Medical Research Council of Canada was the primary funding organization for research scientists, and back then you could have a success rate between 30 and 40% of the grants that were applied for.
What does it look like now?
Now it's like 18. Sometimes it goes down to as low as the low teens.
Wow.
As high as up to 18. So there's just not that money that we used to have. But on the other hand, there's way more opportunities for people outside of academia. So that's what's really changed over the last like 20, 30 years.
Yeah, that was actually one of the questions I had was sort of, did you consider other pathways? Did you look at medicine, like clinical medicine, industry, biotech? And it sounds like back then, like if you're in grad school, really the pathway was that you would go into academia and become a professor.
Academia or medicine. Medicine. I would say a number of students in the lab went into medicine, either after a master's or some of them after a PhD. That was the other. So there were a number of students that used that pathway as well. But yeah, beyond those two, it changed a bit by the time I had finished my postdoc. So at that point, there was pretty much the beginnings of a robust biotech industry all over the place, particularly in the Bay Area where I was. There's a number of bigger companies that were starting. So that was an option. I was really never really entertaining that. I was really thinking more about going into academia.
Gotcha. Yeah, and I guess I have a lot of questions about a postdoc. As an undergrad, we were like so far removed from it that it doesn't quite make sense exactly what you're doing because you already have a PhD.
Yeah.
But could you talk about sort of how that helped your career?
Yeah. Okay, so first of all, postdocs are the best time of a scientist's life. So this is really true because you get this opportunity to just do research. You're doing research. You can work so hard at research. You don't have any administrative or teaching responsibilities, and you can just get stuff done. And most scientists that I know really find that was their the best period of the most fun period of their life with respect to research. So the postdoc period is great. It really comes from as universities started filling out their departments as they expanded. There was a time. So, for example, my supervisor, he barely did a postdoc. There's some fields that you don't do postdocs at all. But nowadays, we graduate so many PhDs that we don't have enough positions for them. So they started doing this post-PhD period, postdoctoral fellowships, where they would just do research, sort of get their research programs before they start joining a university. So it's really just an opportunity to really find out what you want to do in life with respect to research, so that then you can go to university and say, here's my program. Right. I'm going, and you can see 5-10 years down the road what this person's going to do. So it really adds, that extra experience and that extra time to really hone what you want to do.
Right on. Yeah. What did that look like for you? What was your research interest? What did you sort of hone in on during that time?
Yeah, so interesting question, because I looked at a lot of different types of model systems to look at development of the nervous system. And I was really interested in how the brain wires up, particularly how neurons grow and find their eventual targets. And there was a number of model systems that people had been using at the time. And in retrospect, maybe I didn't pick the best one. I picked a system that I thought was going to be really valuable to me for the questions that I wanted to answer. But I really probably didn't anticipate the way that science was going to go with respect to genomic sequencing and the use of model systems where you could alter gene function easily. So I actually picked a system, Locus, and it was a great system at the time for me. It was I was able to use this to look at individual neurons and see how they grow inside an animal. But they're a very difficult organism to use genetically. They have a huge genome, bigger than ours,
Wow.
and way more chromosomes, whereas things like C. elegans and Drosophila have a small number of chromosomes. So I really enjoyed my time. I started to realize, like 10 years after working with these animals, that and you start to get grant reviews that suggest they're not the organism they want to see anymore. You start to realize that you have to start thinking about alternative ways of doing your research. Yeah.
Interesting. And so Locus just like happened to be, the model that you picked, or was there anything specific about them that made them easier to work
because at
with?
the time they were, I wanted to work with a simple system because I was very interested in understanding how neurons grow at the cell biological level and how they interpret guidance cues. And at the time we didn't have, again, the genomic sequencing so that we could identify guidance cues that easily and find out where they are in the organism. So you had to do a lot of sort of observational science.
Right.
And the beauty of the locus was you could actually, in these locust embryos, you could open up their nervous system. And this was sort of cutting edge at the time. I could label individual neurons and watch their growth cones grow in the organism, which nobody could do that at the time. So it was ideal system for that type of work.
Right. And what kind of staining were you using to like look at these neurons?
So I was using, I was labeling their membranes with these stains called dye-I's and dye-O's. These are just sort of lipophilic dyes that incorporate into the cell membrane. So they would go all over the cell, so you could see their individual filopodia. And it was using this system that, in collaboration with a lab just above me, Dr. Corey Goodman's lab, we identified how growth cones responded to some of the first semaphorins.
Right.
So we were involved in identifying the first semaphorin in invertebrates. And so we used my system to actually assay, what are these proteins doing?
Very cool.
We had no idea at the time that they were really quite repellent proteins.
Interesting.
So it was an interesting time to actually be working with locus.
All right. Yeah, that research specifically with semaphorins comes up in like my developmental neuroscience courses all the time, and biology 458 and others. And so your name always shows up, and I'm like, oh, I was stopped by Dr. O'Connor.
Yeah.
That
was so it's weird how science goes sometimes. We had Jonathan Raper, who was, I think, at the University of Pittsburgh at the time. He had identified an unknown protein that collapses growth cones in a in vitro model. And I was working with another postdoctoral fellow, like I said, in Corey Goodman's lab. And he had identified, he had sequenced a protein. He had found a protein, but he didn't know its function.
Interesting.
And so he and I worked on trying to identify its function. It was hard to interpret based on the phenotype. We used antibodies to block its function. And then as Dr. Raper started to get some sequence of his protein, it turned out they were homologous. And that's how we stumbled on like the first semaphorin.
Wow. And would you say like that's the biggest sort of research contribution of your career? What would you identify as like, where did you push the field the most?
Yeah, I think so. So I think there was a couple of things that we did we really provided some of the first observations on how growth cones respond to guidance cues, because again, we could visualize these growth cones. And in collaboration with Dr. Mark Kirshner's lab, with James Sabry, who was a grad student at the time, we started to look at the cytoskeleton inside growth cones, and again in vivo. And nobody was doing this at the time, so we started to come up with models of how growth cones can respond to guidance cues. So I think that, and I think some of the more recent work that we've done, again with semaphorins, I really love the study that my student did that showed that semaphorins in the spinal cord prevent the early entry of sensory neurons and sort of forces them to go up and down the spinal cord, which makes sense evolutionarily, because you want to use all your spinal cord rather than them coming in and penetrating. You want to make them go up and down and use all the territory they have there. So that was something that we did back in like 2010 or so. And then the most recent stuff I'm really excited about, because we've been looking at things like semaphorins and some of the other guidance proteins in the adult. And it turns out when you remove these proteins in the adult, so not during development, so you're not having an impact on development, when you remove them in the adult, neurons die. And we're not sure why. So we've seen this now with 3 or 4 different guidance proteins. It doesn't happen with all of them, so we screened over 40 of them. But some of them, when you remove them, you have neuronal death. So we think it might be a mechanism of neurodegeneration, for example.
Interesting. So they're neuroprotective in the adults.
In a way, they're neuroprotective. We think they might be important for the structural maintenance of a neuron.
Interesting.
And we think once you start to break down that structure, you don't maintain signaling between neurons. So we think that might be the key. Now, we haven't been able to test that yet. And this work we've just published in the last couple of years.
Sounds fascinating.
Yeah.
Where do you see sort of developmental neuroscience going from here? Like, what do you think are the biggest unanswered questions that you see we might get an answer to in the next decade?
Yeah, that's a good question. I really do feel that there's not a lot of emphasis on development anymore. The Canadian Institutes of Health Research, for example, they actually eliminated their developmental panel.
Okay.
So there's not. I do think people. So one of the questions I still think is unknown is, how we do set up our circuits. So some of the things that I used to work on, and I'm still very interested in. I think we have a good idea of overall the proteins that are involved. We don't really understand the cell biological mechanisms that make growth cones. They have to travel over such long distances, and they respond to so many different cues. We don't understand how they can keep changing their sensitivity from one cue to the other. I think that's a huge question in the field.
Interesting.
There's still, we have people in our department now who, for example, Dr. Brett Hilton, who's recently arrived, and he's looking again, going back to sort of, can you recapitulate the developmental programs in mature neurons to see if we can make them grow better. So I think this is still an area in regeneration that people are still pretty excited about.
So it sounds like we're sort of looking at now, how do we, I know SCI is a very interesting area of research right now with spinal cord injury. TBI is how we can regenerate neurons.
Yeah, so it's always been a big aspect of the development. So how can you apply what you learned from just looking at development of the nervous system to, therapeutic approaches for things like TBI regeneration? Yeah.
Gotcha.
I mean, I think in TBI we're still at the stage of collecting data, still at that observational stage. I think it's going to be a very exciting area to, treat at some point. I mean, of course, the obvious treatment is don't bang your head, but
Sure.
prevent. Maybe you shouldn't go into football when you're 5 years old or something. But so, but yeah, I've read a bit into the field, and I find it really fascinating. All the secondary things that happen are just amazing. I think if we can intervene in those things, that'd be great.
Yeah. I want to ask sort of more generally, what do you see are the biggest challenges when you take on a research project? What is your limiting variable? What makes it more challenging than, a career in industry, for instance?
Yeah. So there's a number of things that are challenging. so when you become a research faculty member, it's very interesting that you go from this period, like I said, being a postdoctoral fellow is one of the best times in your life, where you have no obligations to teaching and administrative work. And then you come to a university and you have to set up a lab. And so there's a couple of things that that you're very aware of. One, you have to get funding, and two, you have to start managing people. So one of the things we don't really get trained with at any part of our career is that we don't get trained in things like accounting. We don't get trained with how to manage people. And these are key elements of actually running a lab, right? So you're a bit like an entrepreneur. And there's always that pressure to get money. So you have these three things that are always weighing on you. And so the administrating the lab, personal interactions with people, how to run people, and getting money for the labs. So those are the challenges. And one of the things you realize over your career is that a lot of science is failure, and it should be. And I think that's a good thing, right? Because I think that our hypotheses shouldn't always be correct. It always seems that they are for some reason, but really you should have a lot of failure. And science, that's the way science is. A lot of your experiments fail. And I tell students that to really be successful, you just got to keep working at it. There's just this magic period that you go through where you start to have things that work properly. So you have a lot of failure at the actual experimental level. And then as a research scientist, there's a lot of failure when it comes to publishing and when it comes to getting grants, right? And again, that's just the nature of the endeavor. So granting is, like I said, the success rate is very low. Publishing papers is I think students find it traumatic often. There's because you get a lot of rejection. So you get a lot of rejection over your career, but it's getting those papers and it's getting those experiments to work. That's when people really, that's when they really enjoy their science. And that's why I think it's important when you're running a lab, when you have those successes, you really have to reinforce them with the lab.
Right.
And make students understand how worthy they are for the whole endeavor.
Very cool. So what I'm hearing is like sort of the administrative side of things, the leadership, all of that is sort of outside of the typical research forte.
Yeah. So you have to sort of pick that up, and you'll see people run labs very differently. And the good thing about young faculty nowadays is that they share information. They share procedures, they share principles on how they do things, right. and that's really helpful. Yeah.
Tell me sort of a little bit about the work-life balance in academia, because you mentioned that in order to be successful, there is that demand to just give it all of your time. You mentioned earlier in our courses that you were like working weekends as a young faculty member, with your family and all that. So what does that look like, and how do you balance it?
Yeah, that's a challenge. I have to say that I even see it with young academics today. It's hard to maintain that balance. Science can be hard sometimes, and it takes a lot of work. And so it does mean that you're going to be working some nights. It does mean that you're going to be working some weekends. Particularly as you get your lab established and you instill the type of, what's the word I need here?
Sort of in the new early stages.
Yeah, you instill like a lab ethic on
a culture.
A culture. Thank you. Yeah. So you instill the culture in your lab. That's necessary to be successful. And so it's helpful to have, I was very fortunate to have an understanding wife at the time who, she put up with a lot of stuff where I was working. And so it's helpful to have that. I think as you grow older, you start to establish that work-life balance. I think I started to, as I got older, put aside things I would do work, try and be more efficient at my work, and made sure that I provided the time for my family, particularly as the family are growing up. So I think it's still a challenge. I see Faculty are dealing with it probably better than we used to in the past. I think people are more aware of it. So, we don't have any expectations of faculty to be here at all hours or anything like that. So I think the perception has changed.
Right.
But we still see people that are having a hard time being challenged by this. Yeah.
Yeah. Sort of on the flip side of that, what advice would you give to undergrads maybe watching this right now or in the neuroscience program who maybe had some courses with you? If they're interested in a career in academia, what should they be looking out for? What's something that maybe surprised you?
So a lot of them I've seen are already knowledgeable in the sense that they should try and get as much experience as possible. And I wish I could take tons of students in the lab. It's just I wish all the labs could take tons of students because there seems to be a real desire by undergraduate students to get that experience. And I think that number one, that's great. Number two, if they want to go into this, I think being able to read a lot, to me, that's a key. If you can read and if you're happy writing, these are definite skills that you need for being successful in research. And then once you start to read and you start to understand the fields, you start to understand like where are the centers of excellence in your desired field. And certainly we have some great schools in Canada, but we're a fairly small country. So you start to identify the big schools in the US as well. And so if you can start to set your goals pretty early, you can start to identify where you would like to do the research, in the questions that you're interested in. And I've seen just in the last few years a number of students from either the behavioral neuroscience program or cognitive science. I've seen people that have really at a young age identified where they wanted, what they want to do, and they're getting into good schools in the States. And to me, that's a good sign. So be aware of what you're interested in, read a lot, and if necessary, you wouldn't necessarily be doing this at an undergrad, but be prepared to write a lot too.
I just want to end off by saying I know how instrumental you've been to creating the undergraduate program here at UBC in neuroscience.
Yeah.
So from all of us over at the UNA, we appreciate those efforts, and we wouldn't be here without that, and I wouldn't get to learn all this cool neuroscience.
Yeah.
Thank you.
Thanks, and I want to give credit where credit is due, and certainly Stephen Barnes was instrumental in putting this program together, and having the input from both Vanessa Ald and Lisa Galea. It was really the whole group effort that put this together. So
awesome.
That was great.
Yeah. Thanks again for your time.